I'd like to thank the organizers for inviting me to speak to you today about my work. I do have to point out I'm at the Medical College of Wisconsin. There are actually, it's okay, 2 medical schools in Wisconsin. I get in trouble if I don't, there's a rivalry. So I think that everybody here today understands that one of the central questions in immunology is how do we exist in the context of our enormous bacterial burden, and what is its role in health and disease, and that burden is predominantly in the intestinal microbiota. So we realize the bacteria manipulates the host response to its own advantage, and I think we'll hear more of that as this meeting goes on. Conversely, the host also manipulates its bacterial composition to its advantage. And the outcome of the competition between the host and the bacteria is what shifts the balance between homeostasis and injury. Now, at our mucosal surfaces, which are open to the external environment, we are in continual contact with microbes, yet we have a very low incidence of infection and also an infrequent inflammatory response. And that suggests that we have effective, non-inflammatory innate mucosal defenses at our mucosal surfaces. Beginning this work many years ago, we thought about it in a very simplistic way, which is that a microbe comes in contact with the epithelium either directly or in some way the epithelium senses the presence of a microbe and then generates a local response. The local response tends to include the production of proteins, inorganics, and antimicrobial peptides, and these work together to either eliminate or neutralize this microbe at the surface. 99.9% of interactions at our mucosal surfaces go along this path. There are cases, though, when for some reason the epithelium senses that this is ineffective at neutralizing our microbes, and in that case it triggers local longer-term responses that involve inflammation and acquired immunity. Now, I'm interested in the barrier response, the 99.9% of the interactions, and a large part of that is the production of antimicrobial peptides. In mammals, antimicrobial peptides are made by white blood cells and also epithelial cells that line every mucosal surface. The antimicrobial peptides have broad-spectrum antibiotic activity, and they function by at least making holes in bacterial cell membranes, though there is controversy on if that's exactly how they kill bacteria. They are found throughout the plant and animal kingdom, and although the peptides vary from species to species, the concept of antimicrobials in host defense is conserved and is a very ancient host defense mechanism. One of the major families of antimicrobial peptides in mammals are defensins. It's probably the largest family of antimicrobials. These are cationic peptides. Alpha, beta, and theta have been described. They're characterized by invariant 6-cysteine arrays that are involved in intramolecular disulfide bonding. In humans and mice, there are alpha defensins. They're found in neutrophils and panacells, and they're constitutively produced. Beta defensins are found in almost every mucosal epithelium, and they are both constitutively produced and inducible by bacteria. Now, defensins and other antimicrobial peptides have multiple activities, and they're not limited to their antimicrobial activity. This is from a review article by Bob Lehrer in 2004, and this is a very limited list, actually, of functions of alpha and beta defensins, which includes interaction with complement, opsonization, antagonism of ACTH, chemotaxis of both immature and mature immune cells, histamine release, wound repair and cell migration, cytotoxicity and growth promotion. Recently described antimicrobials or defensins, in fact, act as antitoxins and also to aggregate bacteria. In mammals, as I said, there are 3 defensins. Alpha defensins, which are, see if I can get that to work again, enteric and also myeloid. So alpha defensins are found, as I said, in PMNs and also in panacells. They have this invariant 6-cysteine array involved in intramolecular disulfide bonding. Beta defensins are very similar in their 6-cysteine array. There's just different series of disulfide bonds. And theta defensins, which are right now only found in rhesus macaques, are from the truncation of 2 alpha defensins that form a circular structure, again with 3 intramolecular disulfide bonds. I'm interested in the alpha defensins in the GI tract, and we're not gastroenterologists, or I'm not a gastroenterologist, so I view the GI tract as a tube, starting at the mouth and ending at the rectum, with varying epithelium throughout. The red dots here represent bacteria. We're relatively highly colonized at the mouth, with very low levels of colonization throughout the gut, increasing in the small intestine, and then, of course, extremely high levels of colonization in the large colon. Now, the small intestine, represented by this box here, is, as our previous speaker said, an area of high surface area involved in nutrient absorption, and it is made up of outpouchings called villi and invaginations called crypts. This crypt-villus architecture is lined by a single cell layer of epithelium and made up of a number of different cell types, all of which derive from the crypt stem cell in the neck of the crypt. These include the absorptive enterocytes, goblet cells, which, as we heard, produce a discontinuous mucus layer, M cells involved in antigen sampling. These overlie Peyer's patches, the stem cell, and then at the bases of the crypts are the Paneth cells. So here's a blow-up of the Paneth cells, which are highly secretory cells that originally were described and speculated to function in digestion, which ironically is one of the few functions that Paneth cells do not have. This is an EM generously provided by Andy Wallette. And as you can see, these dense, highly secretory granules that pack the cytoplasm of Paneth cells, this is a very incomplete list, as a matter of fact, of all the different peptides and proteins that have been described as being in the Paneth cell granule. The dominant proteins are alpha defensins. As I said, there are 6 different ones in mice. They've been called cryptins for crypt defensins. There are 2 in humans, HD5 and HD6. Not that we don't know how to count, but the first 4 are found in neutrophils, so these were found later. Cryptin-related sequence peptides are found in mice. Lysozyme, secretory phospholipase A2, Reg3 gamma or HIPAP in humans, Angiogenin-4, alpha-1 antitrypsin, TNF-alpha, IL-17A, MMP7, IgA, and the list goes on and on. So when I began work in this field a number of years ago, we had a lot of ideas about what Paneth cell defensins could be doing. As I said, they're not involved in digestion. They produce a lot of antimicrobials. So when you take them out of the mice and throw them on bacteria, fungi, they kill them. But in vivo, it was a little bit harder to come by evidence of what they did. And we speculated that they could be involved in protecting from food and waterborne pathogens, such as salmonella. They could be involved in maintaining this very low level, relatively low level of small intestinal colonization. Since Paneth cell numbers increase as you move distally down the gut, they seem to be, that would be a useful role. They could act as regulators of the commensal microbiota, or they could have a function in adaptive mucosal immunity, because there are relatively few molecules that function as effectors that don't also have a secondary role in signaling the adaptive mucosal immune response. So to get at this question, we've used 2 different models, one that we developed ourselves, which was the transgenic HD5 model, where we took the HD5 gene, including 1.5 kb of 5' flanking region. It's a relatively small construct, injected it into transgenic mice. We looked at expression of our HD5, and what we found in our transgenic mice, which are represented by the black bars, that we have relatively equivalent expression of all the main products that we tested in Paneth cells, except that we have expression of human defensin 5. Interestingly, human defensin 5 is not particularly overexpressed in this model. It's expressed at the same level as cryptin 4, which is sort of average expressed for one of the mouse panicell cryptins. So we've effectively added one more cryptdefensin, sort of at a normal level. When we look by in situ hybridization, what we find is that we have specific expression at the bases of the crypts, which are where we find panicels. So here we have our transgenic expression of HD5, wild type, of course, none. And then in human, we have a much higher level because HD5 is expressed at much higher levels in human tissue. So what we found is we have physiologic levels of mRNA and protein in these mice. There's specific expression in panicels. And what we found a number of years ago was that these mice had increased resistance against salmonella infection. Our other model is the MMP7 knockout model, which is a model for defensin deficiency in the small intestine. This was done by Carol Wilson and published a number of years ago in Science. Now, the defensin or cryptin gene is a pre-pro peptide. So MMP7 is the processing enzyme for this protein, and it processes releasing this active form, which is found in both the tissue and the lumen of the mouse small intestine. In humans, this is very similar, except in humans it's processed by trypsin, and the unprocessed form is found in the tissue and the processed form in the lumen. So in the absence of this matrix metalloproteinase that is expressed in mouse panicels, cryptins are not processed. So you can see here, this is an aciduria polyacrylamide gel. The smaller bands down here are the processed cryptins in our wild-type mice. In our knockout mice, there's no processed defensin. And these mice or crypt extracts are less capable of killing bacteria. So these mice are also more susceptible to enteric salmonella infection and also less able to deal with exogenous bacteria that are given orally. So we have our complementary models, and I think between the two of them, we're able to say that one of the roles of panicell defensins is in their ability to protect against infectious pathogens, small intestinal infectious pathogens such as salmonella. Then we turned our attention to other possible roles. The reason we were interested in other possible roles is that we have a constitutively expressed antimicrobial, and it seems an unnecessary expenditure of mouse or human energy to be constantly pouring out an antimicrobial just for the occasional incidental pathogen. So we felt that there was much more likely to be a role in homeostasis. So using both of these models, we started to look at the commensal microbiota. And our homozygotes are black, heterozygotes are gray, and the wild type are white in these HD5 mice. And this is just total bacterial numbers analyzed by 16S ribosomal RNA. And we find that it doesn't matter whether we add HD5, we're not changing total numbers in the small intestine, cecum, or large intestine. More interestingly, it doesn't matter if we actually knock out all active cryptins. We're not changing total numbers, again, in any area of the gut. So it appears that panicell defensins are not involved in total bacterial number. However, when we looked at microbial community comparisons, we found something significantly different. We used breeding pairs of heterozygous or partial knockout mice. So we were breeding in our litters both wild-type heterozygote and homozygous mice, or wild-type partial knockout and complete knockout mice. And what we found is when we compared our black 6 wild types to the MMP7 knockout, we saw a significant shift with an increase in the Firmicutes bacteria and a decrease in Bacteroides. Interestingly, when we looked at our HD5 transgenic, we saw a reciprocal shift with a decrease in Firmicutes and an increase in Bacteroides, which we thought was actually pretty interesting since these mice are on different backgrounds. We did this both by, this was done by 454 sequence, not 454, this was done by whole 16S sequencing, and then we did by quantitative PCR, where I grouped the different groups we looked at, making them somewhat similar in color so it would reflect what's going on here. And we saw the same reciprocal shifts in terms of decreasing Bacteroides versus increasing Bacteroides depending on defensin expression. What we also noted was an absence of a particularly interesting bacterial group, and I know you're going to hear more about this throughout this meeting, is in our transgenic mice, in their litters, we had a complete loss of segmented filamentous bacteria. This is a fluorescence in situ hybridization analysis of segmented filamentous bacteria from the mouse terminal ileum. So all the panels on the top are specific for segmented filamentous bacteria, which are these large green organisms that basically directly contact this villus epithelium, which is not so bright here, but you can see it's outlined in these different images. At the bottom is a stain for all bacteria, which are red, so you should see both the segmented filamentous bacteria and any other bacteria in the background. Now, if you order mice from a specific vendor, which we use, which is Taconic, you find segmented filamentous bacteria in abundance in adult mice. In our transgenic mice, however, this bacteria has completely disappeared, even though there's enormous numbers of bacteria still present in the mucus. This is all autofluorescence. Interestingly, in the wild-type littermates, the segmented filamentous bacteria also disappears. And what we learned through a number of complicated experiments was that the bacteria was disappearing in the breeding pairs, and the parents are not transmitting it to their offspring. If we gavaged wild-type mice, these littermates, with cecal contents containing segmented filamentous bacteria, we're able to reestablish this organism in the wild-type mice, but we could not reestablish that in our transgenic mice. So why are we all interested in segmented filamentous bacteria? It turns out to be a pretty interesting microbe. This is an overlay of a specific name for SFB, background bacteria here, and this is the villus. So starting many, many years ago, this has been described as a gram-positive, spore-forming, and uncultivable member of the intestinal microbiome of diverse species, but only once described in humans. It's sensitive to IgA, antibiotics, it directly contacts intestinal epithelial cells, and it induces complex maturation of mucosal immune responses, and I'm sure you're going to hear more detail about that. And also SFB colonization protects from pathogen infection and pathogen colonization. These are a number of interesting references that all speak to this information. So we were interested in learning what this was doing in our animals, and what we found when we looked at T cells from the lamina propria, we isolated them, stimulated them with PMA and anamycin, and analyzed them by flow cytometry. We found that the T cells from HD5 mice lacked IL-17A expression. So here we can see at the bottom by percentage or by total number, HD5 mice, LPLs, have no IL-17A expression. Wild-type mice that lack SFB have no expression. Low levels of SFB lead to low levels of expression, and high levels of SFB lead to sort of standard levels of expression. Our MMP7 mice, which never lose this organism, have reasonable levels of IL-17 expression in their T cells. So that suggests that, although this wasn't the focus, that IL-17 expression in T cells is directly proportional to SFB abundance in the small intestine. But what was more relevant to our interest is that defensins may be able to regulate mucosal T cell responses through their ability to regulate intestinal colonization. So back to this, I think we figured out, we already checked this off, and that pennicell defensins can regulate the type of intestinal commensal bacteria, and through that can regulate adaptive mucosal immunity. This is of particular interest because of the implications for understanding inflammatory bowel disease, particularly Crohn's disease pathogenesis. So again, not to belabor a point, you change defensins, you change the microbiota. You change the microbiota, you change mucosal immune responses. And interestingly, abnormalities of bacterial colonization or dysbiosis is a hallmark of Crohn's disease. Several of the primary genetic defects of Crohn's disease, particularly ileal Crohn's disease, have been associated with pennicell dysfunction. These include NOD2, ATG16L1, XBP1, TCF4, just to name a few. And interestingly, that pennicell defensins regulate the microbiome and consequently mucosal immune responses. Some work by Jan Wekamp a number of years ago looked in patients and found that patients with Crohn's disease also have reduced panacell defense and expression. And they looked at patients with normal NOD2, but still with ileal Crohn's disease, and with the various NOD2 mutations. They found that all of them had reduced defense and HD5 expression. The SNP13 mutation had even lower levels of HD5 expression, and this is the mutation that's been associated with increased or worse ileal disease. Interestingly, the reduction in HD5 expression was irrespective of inflammation, which means it wasn't as a result of inflammation. So that suggests that maybe changes in HD5 could be an inciting event in Crohn's disease pathogenesis, possibly in the dysbiosis associated with Crohn's disease. So essentially what we have, and that what we looked at is genetics, in our case, transgenics, changing panacell antimicrobial peptide expression, or in the cases of IBD, alter intestinal antimicrobial peptides. What I didn't discuss, but I probably may be discussed or may have been discussed earlier today, other things can act to change intestinal antimicrobial peptides, such as inflammation with an influx of neutrophils, which is our other major source of antimicrobial peptides or defensins in the gut. These can lead to changes in the microbiota, and the microbiota changes can lead to changes in colonization resistance, which may render individuals with increased susceptibility to intestinal colonization and infection, or changes in T-cell activation, which could lead to uncontrolled local mucosal responses, such as IBD, or systemic responses as seen in EAE, autoimmune arthritis, asthma. I did want to make one point with regard to panacells and the regulation of intestinal homeostasis. There's been a lot of recent evidence that suggests that panacells not only produce antimicrobials, but also are important cells for the maintenance of the crypt stem cell. So all of these things could impact panacell function, not just defense and production and antimicrobial production, but also whole panacell function in terms of maintaining the crypt. So one could understand why, if you disrupt the function of a cell that is important in maintaining a crypt, well, you're going to have something that results in a collapse of homeostasis at the mucosal surface, leading to dysbiosis and disease. Finally, I would like to thank the people who've been involved in this work in my lab for long-standing research in this area, long-standing collaboration with Chuck Bevins at UC Davis, Nico Bos at the University of Groningen in the Netherlands, and Yvonne Patterson, in whose lab this work was started. The Williams lab at MCW was responsible for doing the flow cytometry, and George Weinstock and Erica Sodergren at Washington University in St. Louis for our deep sequencing, and of course, my funding sources.